Hi everyone, my name is Edouard and I am a researcher at the Division of Combustion Physics in the Department of Physics at Lund University in Sweden. In this presentation, I'm going to talk about exhaled saliva droplets and aerosols measured in time and 3D space. Before starting, I would like to thank the organizing committee who has invited me as a keynote speaker in the laser light interaction with particles in 2022. However, this conference was first planned in 2020, but because of COVID-19, it has been postponed to 2022. So the work we have been actually focused on during those 2 years was to understand how the transmission of COVID-19 from one person to another is made via airborne through aerosols and saliva droplets we emit when talking and when coughing. So this is the goal of this presentation, to do advanced measurement of those particles. Before starting, I would like to thank my PhD students and my postdoc. First of all, I would like to thank Adrian Ross, who has been very active in doing some advanced image post-processing to extract quantitative information. Then I would like to thank Medistiti. He has been focused on the evaporation of water, saliva droplets. Finally, David France has been focused on developing and applying the experimental setups. Thanks to those colleagues, a lot of the work presented here has been very successful. So, before starting the complete presentation, I would like to explain one of the description on what's happening when a person is speaking and coughing. And we have defined 4 types of particles that are exhaled from the mouth. The first type of particles are small aerosols, and those are less than 5 microns. The second type of particles we have been defining are the large aerosols, ranging between 5 and 20 microns. Then we talk about the droplets between 20 and 100 microns. And finally, we define the large droplets that are above 100 microns. And this categorization is what we have been doing over the past 2 years. And we can actually define over time what's happening. So first of all, what is exiting the mouth directly are liquid particles. Then there is an evaporation, a transition from the liquid to the solid particles. Finally, what remains and suspended in the air are mostly the smaller and large aerosols, which are solid particles. The large droplets have mostly reached the ground. So, in this presentation, we will first focus on what is exiting the mouth. Or let's say we have been developing a laser technique for this. Then we have also simulated the evaporation, the transition from liquid to solid. And this is another section of this presentation. So let's summarize. First of all, I will talk about this transition from liquid to solid particles. Then we will describe how the optimized high-speed laser imaging setup is occurring. After that, I'll talk about the particle sizing and 3D tracking. How can we extract quantitative information from what we have recorded in the lab? Using this data, we can actually quantify the SARS-CoV-2 flow rate. And this corresponds to the 4th section. Finally, I will end up this presentation on mask efficiency. How different types of masks are blocking those particles. In this section, I'm going to talk about the transition from liquid to solid particles. We have been using this acoustic levitator where an in-air acoustic streaming and an outer acoustic streaming is maintaining droplets in the air at the same location. By illuminating these droplets and imaging it with high resolution, we can analyze how this droplet will lose water, evaporates, and reach a solid particle. One thing to remember is that the amount of water in saliva is around 98%. A large amount of water is evaporating, the solid particle is becoming much smaller, and this is what we are analyzing here. So as we can see here, on the surrounding, this corresponds to the dimension of the liquid droplets. And now the resulting solid particles has this dimension. If we look at different types of these residues, we can actually see how they look like. Even though they are deformed, maybe due to the acoustic levitator, there's still an impact of that. We can still analyze how the surface looks like and how light may be reflected on this surface. And this is interesting because it may protect virus inside this solid residue, especially if UV is illuminating it. So there may be some reflection out of it. So there is an interesting point to analyze there. Then we have done this experiment where we have different size of droplets and we can see this transition. We have 606 micron solid particles that was generated from originally 1.9 millimeter droplets. Now we have put some dye in the saliva, and this dye was fluorescing. And what we can observe here is that fluorescence, after the excitation of 450 nanometer using a laser, we can see that fluorescence is maintained very strongly in the solid particle. So if this is maintained, we can assess that the virus is also staying inside of those particles, because the virus is 10 times larger than a fluorescent molecule, knowing that the SARS-CoV-2 is around 100 nanometer. If now we are using how the droplet is becoming smaller and smaller, or let's say the final solid residue, once again in comparison to the first droplets. And in this case, we have a higher resolution camera and we can look at smaller droplets. So if now we apply this, or we do those experiments of a shadowgraphy to different persons, for example, here we have 5 men and here we have 5 women, and we can see that statistically the size of the residue is 26% of the size of the initial droplet. And if this one is 21, this one is 22, and so on, right? So if we statistically check out what is the percentage of the final residue, is 23.3% from those measurements from the initial size. So by knowing that, we can simulate and stop the simulation to reach 23.3% of the initial diameter. This is a result of the numerical simulation shown in red, while the experimental data is shown in blue. And we can see here it's linear because we are looking at the surface area. So there is a linearity of the evaporation. And if we look at the larger droplets, we are also shadowgraphy here, then we can see exactly similar than the experimental data. So this model is well validated. And this model was based on the evaporation model from Arbrazon and Sirigano from 1989. And in that case, pure liquid was taken into account, while we have been taking into account a big component droplets, water plus salt. And we have figured out that the 6 grams of salt per kilograms is used in the simulation to have this nice similarities result between experiment and numerical results. So using this model, we have looked at how different droplets of diameter, 20 microns, 50 and 100 microns, are becoming smaller and smaller, how water is evaporating over time, right? different size, different temperature. In this case, we assume an initial temperature of the saliva at 37 degrees, while the relative humidity of the air is 40% here, so it's pretty dry. And we can see that at 20 degrees, in 0.6 seconds, a 20 micron droplets become 4.7 microns, so it becomes an aerosol, extremely fast. The 20 microns droplet coming out of the mouth directly, while 50 microns will be 3.6 seconds, 100 will be 14 seconds. However, one thing to remember is the airflow coming out of the mouth is reaching very close to 100% relative humidity. So we have made another simulation here where we are assuming 80% relative humidity, right? And in this case, the 20 microns at 20 degrees is reaching 2 seconds instead of 0.6. It's more than 3 times slower in this case, right? If now we look at the 50 microns, at 20 degrees, from 3.6, we are reaching 12.4, so nearly 4 times longer. If now we are considering the 100 microns droplet size, it takes much longer to have all the water being evaporated due to the larger volume, the droplets. So there is a high probability in this case that the droplets will fall on the ground before traveling further away. And we can see that for the 20 microns, we are reaching nearly 50 seconds in comparison to 14. Right. If now we have applied those simulations and we assume a person of 1.80 meters high, we can see how long it's gonna take for the droplets to fall down. And for very small droplets, we can see that it takes very long time before reaching the ground, right? While around 80 microns, it's reaching the ground just at the time it becomes a solid particle. While all the other larger than that will not reach, will reach the ground before becoming a solid particle. And if now we look at a 40% relative humidity in comparison to 60, we can see a shift of the time it takes. If now we assume an airflow, not just a drop falling down, but an airflow, and still doing a simulation in 1D, we can see how those 100 microns droplets are traveling before becoming solid residue, the 50 microns and the 20 microns. And we can see that the 20 microns is maintained same height, around 1 meter 20 away from the person, while the 50 microns is away around 1 meter away, but it's falling down. There is a lower probability. At this height, around 80 centimeters, it can be caught by another person. While the 100 microns, around 50 centimeters away, it can be breathed. Otherwise, it will fall on the ground. So these are just some simulation on what is happening in 1D. After the appearance of COVID-19, a very strong interest was to image particles that are exiting the mouth. And this was done in 2020, mainly by basically using a mobile phone, such as an iPhone. And what about the frame rate of a mobile phone? What about the exposure time? Is exposure time short enough and the frame rate fast enough to be able to image and count each of the particles exiting the mouth? Probably not. So in this section, I'm going to talk about an optimized high-speed laser imaging setup where each particle can be detected and where there is no overlap from one particle over the other. I'm going to start with a description of the experimental setup as shown here. We're using two lasers, CW, one at 450 and one at 532 nanometers. Those are combined using a dichroic mirror, and we have a beam expansion. Then we have a top hat, laser profile, and cylindrical lenses allowed to generate an illumination volume between 15 and 120 millimeter. Then we're using two cameras. High speed. We're recording 15,000 images per second. In addition, we are detecting in the semi-forward direction, 52 degrees. And we record over 2 seconds. And we can detect the particles out of the mouse. This is a picture of the experimental setup I have just described. And we can see here the alignments of all the optics leading to a wide planar imaging. This illumination is then following this distance and being detected by the two high-speed cameras. Those cameras are Phantom's VEO 710. If now a person is located here and coughing in this direction, then all the particles crossing the wide light sheets here will be detected. And this wide planar illumination is located only at 2 centimeters from the mouse. So nearly all particles are being detected. We can see another pictures from here where the wide light sheet is going in this direction. And now we can see again the semi-forward detection of the scattered light. This corresponds to 52 degrees, and there is a clear advantage of this configuration. Here it's shown a backscattering, semi-backscattering detection. And then we have it a perpendicular detection where now we can detect a little bit more those aerosols suspended in the air. And if we keep going in the forward, semi-forward scattering detection, we can see that we have a much stronger signal. So there is a clear advantage of this detection configuration. And these are some examples of the results we can see from camera 1 and camera 2. There's a strong signal of the particles coming directly out of the mouse, and they are crossing the 15 millimeter. Now there is another advantage of the setup, where we can speak and cough from the top. And the advantage of this configuration is that we are crossing a much longer distance than 15 millimeter. We're crossing here 120 millimeter. We can see here the configuration without changing anything on the setup. And the width is 15 millimeter. So basically, we're not detecting all the droplets in comparison to the other configuration, but the one we are detecting, and we can track the droplets. So these are examples of the results shown on camera 1 and camera 2. And we have a high contrast detection of the droplets. Have a look again. Right. And using this data is very advantageous because now we can extract some quantitative information. And the first quantity that is of interest is the velocity. So here's a movie where the velocity is tracked, going from around few meters per second down to 5 up to 20 meters per second, especially when the coughing is occurring like now. The fast particles are going around a single line, while other particles that slow down are moving in different directions. This is not a straight line displacement of the particle with different speed. Now there is another quantity that we can extract, which correspond to the size. So as defined previously, there are different size, and we have defined small aerosols less than 5 microns, larger aerosol between 5 and 20 microns, droplets between 20 and 100 microns, and finally the large droplets above 100 microns. So we can see here a classification of the size of the particles exiting the mouth. So this is another quantities of interest. This is exactly the same coughing event than previously shown. So we can see this classification of the particles. So there are two questions remaining. How did we manage to extract the size of the particles so that we can actually categorize them? As well as shown in the previous movie, how did we manage to extract the velocity to track those particles in actually 3D to obtain the correct velocity? This quantification is going to be explained in the next section. I am going to explain how we did the particle sizing and the 3D tracking. For particle sizing, we have been using a high-resolution telecentric objective fixed now on camera 1. In addition, we have been using a red light around 650 nanometer to do this high-resolution shadowgraphy in camera 1, while camera 2 will detect the light scattered within the wide laser planar illumination. If now we used this high-resolution test target, we can compare camera 1 and camera 2 resolution. Around this number 5, for example here, we have 32 line per millimeter that we can clearly see in camera 1. While in camera 2, here, we have this number 1, this is only 2 line per millimeter. So camera 1 is capable of resolving droplets down to 20-30 microns. This is an example where a droplet is imaged with camera 1. In focus photography, light scattered, out of focus. And we can see on the right corner, droplets crossing the wide laser planar illumination. So now we are trying to figure out what is the amount of scattered light for a given size. And to do that, we are using the Lorentz-Smith theory. Here is two examples. One is for 50 microns and one is for 10 microns in order to obtain a calibration curve that will be experimentally validated. So using the Lorentz-Smith theory, we can also consider this 52 plus and minus 4 degrees, which correspond to the detection of our objective. So here we have our two laser illumination, with the blue one at 450 nanometers, the green one at 532, and we average with this light blue. In this case, we just put a sum of them. By doing so, on all diameters, we can have this calibration curve at this angle of detection. One thing I would like to highlight is what about if we detect at 90 degrees? It's very commonly done. In this case, we have another calibration curve where it's much lower. Around 100 microns is 30 times lower. Around 20 microns, we're 20 times lower. And around 5 microns, we are 10 times lower. So the light intensity detected in the semi-forward direction, once again, is much higher. So we can detect much smaller particles. Right. So this is an advantage of the setup we are using here. And by now, showing this high-resolution shadowgraphy, we can see the link between the calibration curve with those droplets. And we can figure out that camera 2 will size properly the droplets by combining the Lorentz-Smith theory curves and the calibration done experimentally using high-resolution shadowgraphing. Now is an example of the results. We can size droplets exiting the mouth. And we can see the diameter increasing, so we know the number of the droplets and the size. And at 70 milliseconds, many droplets are exiting. In this example of coughing, the larger number of droplet diameter is in between 7 and 10 microns. For 3D particle tracking, we have done that in 3 steps. First, we had to find the pixel coordinates related between camera 1 and camera 2. Then we did a triangulation to obtain the 3D coordinates. And finally, we did a tracking of the particles. For the calibration along the Z direction, we have been using this checkerboard target that we have been moving along Z. Certain images have been recorded both by camera 1 and camera 2. 4 images are shown here, recorded by camera 1, and we can see the local coordinates X and Y shown here. But what we want to obtain from those images is the global coordinates over X, Y, Z. To do that, we have to deduce a transform function shown here. A is a polynomial transform coefficient of the third order equation. And there are many A's that need to be found out. So this is applied on both cameras. Additionally, this is applied for each recorded image. So there are certain functions that are deduced per camera, and they have been deduced at each Z position. So once all coefficients are deduced, as mentioned previously, we use the transform function to deduce the 3D global coordinate. So this is the local coordinates, as explained before. We apply the transform function. Now we have the global coordinates. For example, here we assume this pixel, and we can deduce these 3D lines. And this is shown in red because it's a camera 1. So we have one of them. And if we apply it on all pixels, then we have many lines. And this has also been applied by camera 2, which correspond to the purple lines. So these are the 3D lines for all pixels, and it has been calibrated. Now we are going to talk about the triangulation to obtain the 3D coordinates. So this is an example of a raw data recorded. Then we do a thresholding, so all pixels below 25 count are rejected, and intensity above that correspond to particles. After the thresholding, we have the data as shown here. And we can assume 3 dots, so 3 particles here. And the question is, which one of those 3 correspond here? We need to find a relation between them. And to do that, we are going to use 3 lines correspond to those 3 dots using the camera calibration mentioned previously. So in this case, we have these 3 red lines for each of the dots, while camera 2 will have all options. And at the location where it's very close from each other, then we can deduce position of those 3 particles on camera 2. So using this data, we can both find out in 3D using the triangulation, as well as the position of droplets on camera 2, which one are related between camera 1 and camera 2. Now the last point is to do the tracking of the particle. And if we assume one particle here, we need to figure out what will be the next particle this one is going to move. And to do that, we assume the fastest velocity. So if there is fast velocity, for example, 60 meters per second, this is the area where this particle is going to move. But what will be the direction? So the next image is this one. And there are many options on which one of those particles it may be. And so there are several options. And if we keep tracking, then now we can not only take into account the velocity, but also the acceleration. So using the acceleration, we have a smaller area. And if we take another option, then there are less and less possibility of this particle to travel. And if we carry on, we can see that along this line, there is a very high probability that this particle moves in this direction. So I'm going to show it again. First image, second, third, and so on. And now we have all those options, and we can deduce from those scores which one is the most obvious. And as shown through all other possibility of displacement, this one of score 9.7 is the highest probability. Now the 3D particle tracking has been done, and I'm going to show a movie of how this 3D has been applied. We can see that the particles are going up to 25 meters per second. The slowest are below 5 meters per second. If the number of particles is known, as well as their size, and this is recorded at high speed, for example, 15,000 frames per second, then it's possible to do a quantification of SARS-CoV-2 flow rates. Here's an example. We have the count of particles here and their size. Using their size, we can find out the volume of each of those particles that we multiply by their number. And we find out the final volume of saliva being ejected over time. Here we have a large peak, and this peak corresponds to this one. And it's multiplied by large droplets, around 80 microns. However, if now we multiply this other high peak, but only with aerosols, around 10-15 microns, then it leads to a small peak, as shown here. If now the concentration of SARS-CoV-2 in saliva is known, we can find out the flow rates of SARS-CoV-2. To find this concentration, we have been using those 5 articles, where 76 persons have been investigated and their concentration of their saliva has been deduced. There are large differences of the concentration to those people. Note that this has been measured when having COVID between the 1st and the 5th days, the 5 first days. If we look at the super spreader, the 25% people spreading the most, this is above 10 to the power 7 SARS-CoV-2 virions per milliliter. If now we want the median level, so this corresponds to a concentration of 10 to the power 6 virions per milliliter. For the study we are doing, we are assuming 10 to the power 7 and investigating, in this case, super spreader situation. So by multiplying the volume defined previously by the concentration just mentioned, we have here the virions in each frame. Now we have applied this when saying protect better against COVID at a level of 85 decibel or by yelling it at 105 decibel. The P of protect is emitting up to 100 virions. The word better is also increasing this number of virions. For the word against, there are nearly no virions being ejected. And for the word COVID, the V emit particles. If now we are yelling this sentence, once again, the P of protect, and a lot of virions, in this case above 150. The word better for the word against, large droplets in this case, a few of them. And COVID, the V is responsible. Those graphs are the result of the two movies shown previously. And in yellow up there, we have the number of virions per frame, while in purple we have the virions flow rate, the number of virions per millisecond. And we can observe that the maximum rates correspond to 23 virions per millisecond, and this is for a few droplets of 85 microns, fairly large. And for yelling, we have also large droplets for these high rates of virions. However, we can see several small peaks. And those peaks are more for aerosols that contains less virions, but for smaller particles that may remain longer in the air. So we have here 270 virions, while here we have 159. And this shows the difference between yelling and speaking. Using this technology, we can find out how virions are emitted over time. In this section, I'm going to talk about mask efficiency. And for this work, we have changed the wide planar illumination into a thin laser sheet. And by doing so, we could detect down to 1 microns particles instead of 3.2. And this at 50 microseconds detection. However, one thing to mention is for the wide planar illumination, we could do both a 3D and correct particle sizing. While if we use a very thin planar laser illumination, then it was difficult to actually size properly the particles due to the Gaussian light intensity of the laser sheet. However, with this configuration, we could detect the large number of particles and aerosols being exhaled from a cough. Here is an example of 2 persons. While person 1 is reaching above 12,000 particles, person 2 is emitting close to 3,000. So person 1 is emitting more than 4 times. This is a mask of type 2R. This mask is now a standard mask. In this case, the number of particles crossing the mask is much lower. We can see from those measurements that we were reaching less than 200 particles. This is showing the distribution of the number of droplets and aerosols in space. Here, it's a case when we don't have any mask from the previous curve I've been showing. Now the type 2R. We can see improvement with the type 2R mask. If now we are using those type of masks and we fix them vertically, we illuminate them on both sides, as shown here, using the same high-speed cameras than previously used, we can actually analyze the interaction of the droplets and ligaments coming out of the mouth through the mask. And we can image both sides, inside and outside. We have those saliva droplets impacting inside, while outside we don't see anything happening. And after the saliva droplets, we have been looking at saliva ligaments, which can be exhaled when sneezing, as well as when coughing. We can see those impact inside the mask of type 2R without seeing anything outside the mask. The FFP2 mask, once again, we can see saliva droplets impacting inside, as shown here, as well as saliva ligaments exhaled when sneezing. To conclude this presentation, I would like to make a few points. First, it's very important to use high-speed imaging, above 10,000 frames per second, in order to track particles exiting the mouth directly. Then, by using a laser illumination and detecting the forward scattering direction, we can detect the small aerosols. Using two cameras, we can do a 3D tracking. And if now we use a high-resolution shadowgraphy, we can deduce the size of those particles. Using this data and knowing the concentration of SARS-CoV-2 in saliva, we can deduce how SARS-CoV-2 is being emitted over time, the flow rates. Using this input data, it can help to understand better how COVID-19 is spreading in between people. I would like now to thank people who are organizing this conference. Then I would like to thank all of you for your attention.